Hello, students. I hope you're doing well. So I've never done this before, but just the other day I decided to order the textbook, the OpenStax digital textbook in hardcover form. It was like, I don't know, 20 something dollars. It was really cheap. And I didn't know what I was going to get. I've definitely gotten digital textbooks before that are just come bound in two rings, so like loose leaf. But this is a nice textbook. I was pretty impressed. And so I was just messing around looking at it. And we are in Unit 3. And I wanted to talk about this is section 9.1, the structure of DNA. In the textbook, it's page 201. And so we're thinking now about a pretty hard topic, about the structure of DNA. What's the molecular structure? And we just touched ever so briefly on chemistry at the very beginning. And so if you want more feedback on that, I'll post a video that is called A Biologist's Interpretation of the Periodic Table. That's my take on what you need to know about the periodic table for a biology class. So we're looking here at figure 9.3. And what I want you to think about is that when you're reading, it's not just about reading the words, right? It's about taking it in and absorbing the information. So figure 9.3 here, we have two figures, figure A and figure B. And what I want you to think about is how you're always kind of evaluating your own knowledge. You're always thinking about what you know and what you don't know. And so my first question here is like, which one of these is a nucleotide and which is a nitrogenous base? So thinking about layering on vocabulary that you just learned right here in the text. But as you move your eyes over here, ask yourself, oh yeah, this one here is the nucleotide, and these are the nitrogenous bases, okay? And then as you read this page, we're going to learn about how they are complementary bases. And this is called Chargoff's rule. And the basic idea is that through a process where he separated the genetic material, Chargoff was able to determine that there were equal numbers of adenine and thymine and equal numbers of guanine and cytosine, which helped him to infer that these molecules were paired in some way. And so that turned out to be true. Something that we didn't know or understand until much later in empirical fact with the use of electron microscopes. But under our understanding of what DNA is actually on a molecular level is only roughly 70 years old, 1953. So now let's think about figure B and think about figure A separately. So we know that a nucleotide is built up of these three components, and we know that DNA has a phosphate sugar backbone, right? And that something is going on inside of here to cause it to coil. And we see here that there are all sorts of different colors. And so when you're looking at the diagrams, that indicates a different kind of molecule. So as we transfer our understanding of this double helix up into this image, okay, what I want you to think about is this is the ladder. These are the sides of the ladder, okay, over here, the blue, the coiling part. And then in the middle, we have bases. And since they have different molecular structures, it actually contributes to this coiling. And then also we know that here, since there's two different colors in the rungs of the ladder, right, that these are the complementary bases, A with T, G with C. And then when we look over here, we see that when adenine and thymine are connected, they are always connected with 2 hydrogen bonds, and C and G with 3 hydrogen bonds. And as we can look at this basic molecular structure, remember that the carbons are implied by the shape of this molecule, but they're not actually written in to the molecule. So we're not seeing all the atoms that are in these molecules. But some important things to think about are that with every twist and every pairing, the overall length of the molecule is the same. And that helps to contribute to this design. And so then when we're looking up here again, let us think about what we know about phosphate, right? It plays an important role in our energy, ATP molecules. And we see here that this O has a negative sign in the superscript, and that means that this part of the molecule has a partial negative charge right here. And that's important for bonding. And then here in our ribose sugar, we have our implied carbon ring. Remember that all molecules that are sugar end in O-S-E, okay? And then here, when we're looking at this base, right, we can ask ourselves, which base is this? And then as we look over here to the purines or the pyrimidines, we can see that it's definitely a purine, right? And so we can take it even further and ask ourselves, which one is it? Based on the drawing, the way they've drawn this here, and the bonding arrangements, okay? So when we look at this structure here, we have things implied and not implied. So, you know, in the carbon ring, where there's a carbon, there's no C, but where there's a nitrogen, there's an N. So we can use that to sort of compare these molecules. And we can see here that in the adenine, we have a C here, and here it's implied, and here there's an N. And so then we would expect there to be an N right here in this figure. Okay, and then we can also check the bonding. These two lines mean that the carbons or the atoms are double bonded or single bonded. And then we can look at this functional group, and actually the difference between these bases relates to these functional groups and their positions, right? So we can see here that this structure matches adenine. So here we just spent about 5 minutes talking about one figure. And so when I say to you, you know, you need to engage with the material every day and think about what you're doing, maybe you just open the book and you look at a single figure. You don't have to be reading. You can really be taking in the information in a more multimodal way. And then the other thing I was going to show you is, like, when you're reading in this paragraph, it talks about how the two strands of the double helix are anti-parallel in nature, and that is, one strand will have the 3 carbon of the sugar in the upward position, whereas the other strand will have the 5 prime carbon in the upward position. Okay, and so when we take a look at this image, we want to see if we can understand what that means. And also, let's try to find the sugar phosphate backbone, right? And then the bases, and locate ourselves at the 3 prime carbon of the sugar, right? So it has to be within this part. It's not in the middle. That's where we're looking. And then we can see here that right here, this hydroxyl functional group is in the upward position, and over here it's in the lower position. And so there we took one sentence and we just spent 2 more minutes, but we looked more deeply at the figure to understand what's going on here. The last thing I'll note is that these hydrogen bonds are drawn in dotted lines because of all the different kinds of bonds. Hydrogen bonds are very weak, and this is important to keep in mind because the DNA backbone unzips in replication, which we'll read about soon. And if these were stronger bonds holding the bases together, the unzipping would not occur in the way that it does. And so we can appreciate how the sugar phosphate backbone is covalently bonded, and then the bases are bonded with hydrogen bonds, which are weak molecular bonds. Okay, so we just looked at literally one page in the textbook, but hopefully that gave you some insight into the structure of DNA and also how to interact with your reading when you're in this stage where you're thinking about the quiz and preparing your mind and reviewing. Let me know if you have any questions.